Hello, welcome to the first ever webinar by the British Environmental Virology Society, or BEVS for short. This is a re-recording of the webinar that was first presented on the 18th of June. Unfortunately, the recording of the live event didn't work. So Kata and I have re-recorded this so that anyone that wasn't able to attend on the 18th could see Kata's presentation. My name is David Walker. I am an environmental microbiologist at the Centre for Environment, Fisheries and Aquaculture Science. Before Kata's presentation, I just wanted to give a quick introduction to BEVS. So last year, Kata and I decided to start up BEVS after we'd been working together on the VirAqua project, which was a project looking at developing methods for detecting and characterising viruses in the aquatic environment. We came to the conclusion that while there's a lot of people working on viruses in the environment in the UK, it's often difficult to know exactly who's doing what and where. So the aim of BEVS is to bring together scientists and other people with an interest in environmental virology so that we can share knowledge and information and hopefully build collaborations. At the moment, BEVS is just being run by Kata and myself, mainly just communicating through Twitter and by emails. We'd really like to hear from anyone that would like to get involved with BEVS, whether that's by contributing to things like newsletters, sharing job adverts, presenting webinars like this, or anything else you think you might want to contribute. So please get in touch with us either on Twitter or through email. So that's bevs@bevs.org.uk. We've also got a Facebook page that's not very active at the moment, but we plan to be more active on that in the future. Also, at the end of this video, there'll be a link to a survey for feedback on this webinar and on BEVS itself. So we'd really appreciate it if you would fill in that survey. But without further ado, let's get on with the reason why you're all here, which is the first ever BEVS webinar by Dr. Kata Farkas on wastewater-based epidemiology for COVID-19 surveillance.
Hello, good morning. My name is Kata Farkas. I'm a postdoctoral scientist and environmental virologist at Bangor University. And my main focus is enteric viruses in wastewater and in the wastewater polluted environment and their survival, ecology, and quantities. However, most recently, in the last 3 months, we have been started to work on wastewater-based epidemiology for COVID-19 surveillance as well. And this is what we will cover today. So viral diseases, not just enteric viruses, it's not just COVID, it's a lot of different viruses, especially in the last 20 years that caused major global health threats. For instance, the first SARS epidemic in 2003 with over 8,000 cases in 29 countries, that was quite devastating on its own. Followed by 10 years ago, the H1N1 or swine flu pandemic with 60 million cases estimated in over 200 countries and territories. So it was a global pandemic. And what actually helped mitigating and finally stopping this pandemic was the vaccination and the global administration of it. Also, around 5 years ago, we had the MERS coronavirus epidemic, mainly in Asian countries with over 2,000 cases, which is much less than the last two. But actually the mortality rate for this epidemic was really high, 40–50%, meaning that every other person who got infected actually diseased, so that was something very, very major. And also we often forget about this one, the Ebola, which has been around since the 70s, causing sporadic outbreaks and epidemic. And again, just 5 years ago in Central and Western Africa, there was a major epidemic with 15,000 cases, which was very hard to mitigate and actually doesn't receive that much press. But right now there's again a sporadic outbreak in the Republic of Congo, with the number of cases increasing day by day. So that's something to keep an eye on as well. And again, now we have the COVID-19, again, a coronavirus pandemic with over 8 million cases right now, in over 200 countries and territories. So it's a global health threat again. I don't have to tell you about the effects of it and the mitigation. I'm sure you're checking the news and the numbers every day, just like I do. So that is what's going on right now. Actually, what's similar in all these viral health threats, first of all, they are all viruses. They're all very hard to mitigate, or there is no cure for them. There are limited vaccination abilities for them. And what else is very similar to them is that they are present in poop. So actually the infected individuals shed the viruses in quite high concentrations in their feces, which then goes to wastewater collection and treatment facilities in the countries lucky enough to have those utilities. And then what we can do is to use this wastewater in order to receive a snapshot of the public health, of the community health. So what we can do is to collect samples of wastewater, check for the presence and quantity of viruses in them in order to predict or to have a status of an ongoing outbreak at a community level. Because you know it just as much as I do, you can't test everybody. We have limited testing capabilities even now. We just can't test the whole community every day or every week. But what we can do is to see whether the virus is present in the wastewater and what are the quantities. The major advantage of wastewater-based epidemiology is actually the inclusion of asymptomatic and pre-symptomatic cases, which is quite a lot of cases in case of the COVID-19. It is estimated that 80% of all cases is either asymptomatic, pre-symptomatic, or mildly symptomatic. Which means that these cases, 80% of them won't get tested because it's just not severe enough to do so. But if we do wastewater-based epidemiology, as these patients or cases shed the virus in feces, we are able to capture them. And actually this approach, wastewater-based epidemiology, has been in use for poliovirus surveillance in the countries still affected by polio. For instance, Pakistan, they use it as a routine analysis in order to see whether the virus is present in a community. So it can be used. And also there's many data on enteric viruses. So this is, as I said, my main interest, enteric viruses. These are viruses causing gastrointestinal syndromes like norovirus, enterovirus, or hepatitis A and E viruses. And as they resist wastewater treatment and survive in the polluted aquatic environment for a really long time, they are able to cause waterborne or foodborne illnesses. So it is an existing public health threat. And this is why we actually have much data on wastewater quantities for these viruses. For example, in our previous project, the BEVS, which sadly ended 2 years ago, it was a great project, great collaboration. We did do wastewater monitoring for a year at our local wastewater treatment plants in order to capture these enteric viruses. And just very briefly what we found and how this method can be used is that there are certain viruses, for instance the adenovirus or polyomavirus, which usually are asymptomatic in infected people if they don't have any underlying health condition. So probably some of you or most of you actually have either or both of these viruses right now. So these viruses are usually present in wastewater at quite high concentration. They don't show any seasonal changes or daily changes. Whereas other viruses, for instance the norovirus, which is the main cause of viral gastroenteritis globally, do show seasonality. This is why these viruses are called winter vomiting bugs as well, because they usually cause illnesses in the winter months. So these viruses had some spikes in wastewater, and actually most of those spikes co-occurred with known local outbreaks, whereas there were some other spikes where the public health offices did not note any outbreak, however it was clearly present in wastewater. So you can see that this actually can be used as a tool for the surveillance of outbreaks and to capture not known outbreaks where cases are not known by public health. Now it sounds really good, isn't it? You just take some wastewater, check for viruses, you have all these numbers ready. However, it is not as simple as it sounds. And this is what this presentation will mostly be about, is the limitations and the hardship of detecting viruses in wastewater. So in general, for viruses, we have different methods to detect them. In most cases, in the overwhelming number of cases, we use molecular detection, mainly quantitative PCR. However, digital PCR, next generation sequencing, and immunoassays are also in use in some cases, and some other molecular methods like isothermal amplification. But in the majority of cases, quantitative PCR is used, which is great. It's very sensitive and accurate, allowing strain level detection in a few hours. So that's a really good method. However, it only detects a very short segment of the target genome and nothing else. And if you're interested whether you actually have intact and infectious viruses for risk assessment, you can do integrity assays, which means that you eliminate the non-encapsulated viral nucleic acids by enzymatic treatment or with dyes, or you can pull out intact capsids and then quantify the viruses in that sample. You can also use infectivity assay if the virus is culturable in the laboratory. SARS-CoV-2 is actually culturable, so that is doable for that. You can also use electron microscopy and different dyes to produce these really vivid, colorful images of your target virus. And there are some other methods, emerging methods, which may be suitable for wastewater samples to monitor viruses, but these are the main methods used. And actually, so far, there are many studies on wastewater surveillance for COVID monitoring, but actually all of these studies so far have used quantitative reverse transcription PCR, usually in a one-step assay to first reverse transcribe the viral RNA, because this is an RNA virus, followed by a quantitative PCR. There are many assays targeting different regions of the target genome, on the nucleocapsid gene, the spike, the envelope proteins, the RNA-dependent polymerase gene, the RF1, and so on. There are just very a lot of qPCR assays available, which can be used for SARS-CoV-2 in wastewater. The problem with the detection here, if you use quantitative PCR or other types of PCR, is the tiny amounts. So in a qPCR assays, you use usually up to 5 microliter of your sample, which is not a lot considering the volume of wastewater being treated on a daily basis at a wastewater treatment plant. So in order to have a reasonable understanding on viral quantities, you have to concentrate your wastewater sample to detect the virus of interest. And this is where it gets interesting. There are many different methods that you can use. There are very carefully validated methods, which has been used for enteric virus surveillance, as I mentioned before, because it's actually a public health threat. For instance, filtration. What you can do is to use a filter and push through your sample and then capture the virus, which has different charge as your filter material. So kind of absorb your viruses on the filter material. by electrostatic interactions, and then you can elute the viruses later on. You can also use ultrafiltration, which is basically mass exclusion. So you have a filter material with very small pore size, smaller than the virus, and then you just push your sample through by centrifugation or applying pressure, and then your viruses will be retained because those are larger than the pore size of the filter material. You can also use ultracentrifugation if you're lucky enough to have this very sensitive and expensive piece of equipment in your laboratory, because viruses are really very small and they're very light, very low density entities, which means that regular benchtop centrifuge that you use for bacteria or fungi or algae won't work at all. So you need really high forces, like 100,000 Gs, to pellet down the virus. Hence, you need an ultracentrifuge. You can also precipitate viruses, though, if you only have benchtop centrifuges. This is a very popular method as well. You can use, for instance, polyethylene glycol for that. You mix that material with your sample, and viruses and nucleic acids will bind to this material, and then you can centrifuge down the polyethylene glycol with the viruses attached to them. Flocculation is also quite popular. People usually use iron, aluminum, or skim milk to do that, and then once you have the viral flocks, you can either centrifuge or filter them. So these are all very good methods. The problem here is the co-concentration of inhibitors. That you have to be very careful with inhibitors because in environmental samples like wastewater or river water, these type of materials, soil as well, there are a lot of inhibitors, organic matter in your sample with very similar size and charge as your target viruses. And what happens is that when you try to concentrate on viruses, these organic matter will be co-concentrated and highly affect your downstream analysis for viruses. And that can actually result in false negatives. So what you need to do is to balance your viral recovery and the elimination of inhibitors in order to have a really good method that works for your target virus. Now, for the detection of SARS-CoV-2 in wastewater, as I said, there are many international studies available on that matter, and they use basically all the methods that I mentioned so far in order to concentrate the viruses. It usually works with rather low volumes, like 200, 100 mils. Ultracentrifugation, for instance, only works for 11 mils, so that's not quite a lot of concentration. What you can do if you want to increase your sample volume is to combine different techniques. And for instance, if you combine precipitation with ultrafiltration, you can actually filter through a liter of wastewater, which is more meaningful than smaller volumes. Now the question here is how reliable these methods are actually that people have been using for COVID surveillance, because as I said, these methods mainly has been validated for enteric viruses. Now the problem here is that enteric viruses are not like SARS-CoV-2 in there are many features. For instance, enteric viruses are non-enveloped, whereas SARS-CoV has a membrane envelope. Their size is about the same, but enteric viruses are negatively charged at neutral pH and hydrophilic, whereas SARS-CoV-2 is positively charged and rather hydrophobic. So actually, how can we be sure that the method that we use actually able to concentrate and detect the SARS-CoV-2 or any other virus of interest? Now, to study that, actually, there's a group in Australia which tried to work out the recovery efficiencies of the different methods that people have been using for SARS-CoV-2 and other viruses. What they did is that they took a wastewater sample and added a control virus to them. In this case, they used the murine hepatitis virus, which is an enveloped coronavirus. It's very similar. The surface properties are very similar to SARS-CoV-2. So they assumed that the recovery rate of this virus will be very similar to the recovery rate of SARS-CoV-2. And then what they did with the spiked samples, they used different methods: filtration, ultrafiltration, pack precipitation, ultracentrifugation, in order to determine the recovery rates. And as you can see, there were quite differences in recoveries using different methods. Some of them worked much better than others. However, they did find that the standard deviation among replicates were pretty high as well, meaning that there are other factors to consider as well. And this is where it gets a bit tricky, because I said you have to balance out the recovery of the virus and eliminate inhibitors at the same time, but also you have to be careful on the sample type, the volume, the sample properties as well. For instance, if you have a method that works pretty well with a low turbidity wastewater sample, it won't necessarily work that well if you have a higher turbidity sample or a sample with different properties or intake. Also, do you have the equipment available? You need to consider that as well. For instance, most labs do not have ultracentrifugation, or there can be other limitations on availability. How feasible your sample is for mass sampling? Your method for mass sampling, that's a question as well. Because if you want to do a thorough job, you need to take a lot of samples in, like on a daily basis, from different wastewater treatment plants, which can be really time consuming, especially if you have a very difficult method to use. Also, the costs, some equipment or even consumables can be really, really pricey as well. So you have to consider that as well. And also the target viruses. Do you just want to detect SARS-CoV-2, or you're interested in viruses with different surface properties, which you want to co-concentrate? So as I said, there's a lot of things to consider and a lot of things to be careful about when you validate your method to use. So it is not as easy as it sounds. And this is why what I always say, and I think this is very important, is the use of controls. It is not a big thing to do, just to add a virus with very similar structure as your target pathogen to your sample before you process it. So use it as a whole process control. Or you can just quantify indicator viruses before and after concentration, which are usually present in wastewater at high concentrations. For that purpose, you can use gut-associated bacteriophages. It's a popular choice. You can also use extraction control, just to see whether your RNA and DNA are extracted properly. And it is also advised to use PCR control or RT-PCR control. So just spike your sample before the actual quantification. This will help you to assess whether you have any inhibitor present in your sample. So this can be, use of controls can be very powerful. And if you use that quality control measure, you can actually have more confidence in your data. You can actually say that, yes, if I don't detect the virus, it's not because a failure in the process, but it is because it's actually not there. Now, following up on that, the perks of detection, let's see some actual examples on what has been done on SARS-CoV-2 surveillance in wastewater so far. Not including all the studies, just a few that I found important. For instance, this one published and peer-reviewed, which is actually the first peer-reviewed report on SARS-CoV-2 in wastewater. It comes from Australia, the same group as the one that used the murine coronavirus control. It is rather a proof of concept because actually it was done in March, and back then Australia was not really affected by the virus, the pandemic, so they had really low concentrations of SARS-CoV-2 in just a few of their samples when they were quantifying a fragment of the N gene of the viral genome. Again, they used qPCR for that. But they didn't use any control measures or anything like that. It's really just proof of concept at this stage. Now, the Netherlands group, on the other hand, they did a very thorough job, and actually they were the first who posted a preprint on SARS-CoV-2 in wastewater and suggested that it can be used as a monitoring measure. And they were able to detect SARS-CoV-2 before the first cases were noted, suggesting that this approach can actually be used as an early warning system for outbreaks. They also assessed the recovery efficiency using a gut-associated phage. They found 50% recovery, which is rather good. What was really interesting here is that they used the different qPCRs targeting different regions of the genome, the N gene and the E gene to be precise, and they actually found quite differences in the performance of these assays. For instance, one assay suggested that over 50% of all samples were positive, whereas another assay didn't find any virus in any of the samples, which again highlights that you really have to be careful and use validated methods that you know actually works. And with a novel pathogen like SARS-CoV-2, it is possible that one assay doesn't work as well as first they thought. So it is really good approach as well to use multiple targets for one pathogen for SARS-CoV-2 and see which assay works better or how actually the virus is present in the sample. Another example, again, this has been peer-reviewed, the study from Spain, where they quantified the N gene, and again, they found differences in the performance of the different qPCR assays. They also studied the recovery efficiency by adding an enveloped porcine virus and the non-enveloped mango virus to their samples, and they found quite reliable recoveries when they used aluminium flocculation. It seems low. We're talking about 10%, 11% in influent and 3-6% in effluent, but it was quite reliable as the standard deviation was really low. So actually, even though the recovery efficiency seems low, the results you have are actually comparable, so that's a good thing. And what's also really interesting and important in this study is that they did find the virus in a few of secondary effluent samples, but the virus was not quantifiable in the actual tertiary effluent that has been released to the environment, suggesting what we already thought, that as it is an enveloped virus, it probably won't last very long during wastewater treatment. But it is really good to have this assurance from actual sampling and an actual study. Another example here, this is actually a preprint, it has not been peer-reviewed, and here the authors try to correlate the SARS-CoV-2 concentration in wastewater with the local case numbers, which kind of worked in some cases, didn't work as well in other cases, which again suggests that the number of cases are really hard to estimate at clinical level, and maybe those were not as accurate as they could have been. And there's another rather controversial preprint. I would really like to see the peer-reviewed version of it. This is an American study where they did some modeling which actually suggested that SARS-CoV-2 is detectable in wastewater a week before the first cases, which sounds really good actually if you have a week to prepare for the cases. However, the statistics here is a bit questionable, or well, it needs to be double checked for sure in order to believe these models. But it clearly shows that SARS-CoV-2 is again detectable before the first cases appear at the emergency rooms, so that is really important as well. And I'm not going into further details on what has been done. There are a lot of studies now globally working on SARS-CoV-2 in wastewater, and there's an international collaboration. You see the link for the website here, where you can find more information on what's going on. And also in the Teams meeting that was set up for this webinar, there's a file folder where I uploaded all the peer-reviewed papers and the preprints on this that has been published so far, so you can have a good read there as well. Now you must be wondering what's happening in the UK. Do we do something about it? Well, actually we do. At our university in Bangor, we have a grant from NERC COVID-19 urgency program. We take weekly samples at 6 sites in England and Wales, and we detect the N gene by qRT-PCR in concentrated samples using cresphage as a process control and the non-enveloped murine norovirus as an extraction control. We do have some preliminary results, but I wouldn't go into details because the controls have not been tested yet thoroughly, so we don't know how reliable the data is yet. But actually we kind of seeing some effect of the lockdown, showing that the SARS-CoV-2 concentration is going down since the start of the lockdown, but we need to check the results for the controls before we can actually show you something. But we will do that as soon as we have the results. And also other universities are working on modeling approaches, infectivity of SARS-CoV-2 in wastewater, and metagenomics as well to see which strains can be found in wastewater. So there are quite a few projects now. I think right now we have at Bangor 4 different projects working on this, and actually we are hiring. So if you're a postdoc or just finished your undergraduate or postgraduate studies, please get in touch with me if you're interested in working with us on this. And to sum it up, what we know so far suggests that wastewater-based epidemiology can be used as an early detection and monitoring for viral outbreaks, including the SARS-CoV-2. However, there are some limitations in estimating case numbers and knowing the actual viral concentrations in feces. Like we don't really know how the concentration of SARS-CoV-2 in feces changes over time, whether people who are symptomatic shed more virus than the asymptomatic cases, how long they shed the virus. There's just no information on it. If you think about it, we only know about this virus for 6 months, so we just don't have that kind of data yet. What's really cool and important, I think, is the co-surveillance of different health threats, different pathogens, which can be done rather easily by multiplexing your PCRs. In one PCR, you can actually detect up to 5 targets. So you can actually detect 5 different pathogens at once, which is a very cost-effective and time-effective way for surveillance. What's just really important is that you have to have carefully validated methods with appropriate controls in order to produce reliable data that actually answers your questions. And another thing, which is not really the epidemiological side, it's rather risk assessment and health threats coming from wastewater and in the environment, is the infectivity state of the virus in these samples. As I mentioned, it's an enveloped virus. These viruses usually don't last very long in the environment, but again, we just don't have the data on SARS-CoV-2 yet to be certain about it, so this is something that should be studied as well. So yeah, thank you for your attention. If you have any questions, you can use the chat box to ask them, and we will do our best to get back to you. Or you can email me or the British Environmental Virology Society at bevs@bevs.org, or my email is k.farkas@bangor.ac.uk. Thank you for your attention.